It's my pleasure to introduce Xiaokang Lun. Xiaokang comes to us formerly from Harvard University, but recently he started his own lab at the University of Minnesota in January of this year. And as far as we know, he's still there, which means that he survived his first Minnesota winter, which is an accomplishment in its own right. Winter probably ended sometime last week in Minnesota, so we're pleased that Xiaokang can be here to enjoy the warm weather. But more so than surviving a Minnesota winter, Xiaokang is here because he's developed a number of innovative technical approaches that have really pushed the field of single-cell mass cytometry forward. In particular, Xiaokang will tell us today about a novel technique that he developed using DNA barcoding to increase the sensitivity of single-cell mass cytometry. So he's really pushing the field forward with his technical achievements, but I think it's also important to emphasize that he's doing so in service of really important areas of biology. So a major goal of his research is to understand signaling networks and how these interact in cancer, and how we can discover new drug targets using these large-scale data sets he's generating. So please welcome Xiaokang.
Thank you so much. Thank you for the really nice and comprehensive introduction. So yeah, I just started my lab at the University of Minnesota a couple of months ago. So I'm gonna present a work that I did when I was still a postdoc here in Dr. Peng In's lab at the Wyss Institute. And so here I was trying to integrate advanced DNA nanotechnology into the platform of mass cytometry to enhance the sensitivity and multiplexity. So yeah, so for anyone who's not 100% familiar with mass cytometry, it is a highly multiplex protein epitope analysis tool that's currently allowed to analyze there are more than 50 targets at the same time. Those targets can be protein, PTMs, RNA, and some of the metabolic sites. And it's at single cell resolution, high quantitativeness, and also the throughput can be really high as 500 cells per second. In the measurements, we can reach almost 2 million cells per hour in the analysis. So how do we do that is that simply put, we label the antibody that conventionally you can do with fluorescence agent with lanthanide metals, metal isotopes, and then use these metals to label the protein or protein phosphorylation site inside the cells. And this metal test can be differentiated or to read out by mass spectrometer based on the differences of their mass. So you can result in a really sharp peak over here compared to the spectrum overlap that you often see in the fluorescence-based analysis. And those cells are injected into the instrument. There's a nebulizer can help to nebulize the cell into droplet, and there's cells passing through the plasma, form the ion cloud, and going to the top, and to be analyzed. And the readout will be the abundance of each metals, and this can be translated into the protein or PTM abundance per every single cell. And yeah, so there's a lot of advantages using mass cytometry, but it's also some problems we need to solve. And one of these is the abundance, as Galit mentioned in the morning session, that is also really critical to measure those proteins with low abundance, because a lot of them really play an important role in biology. So currently, there's difficulty to measure the low abundance proteins at the single cell resolution with mass cytometry, especially when these proteins are in the cells in small size, such as human immune cells. And another application, imaging mass cytometry, which apply the laser ablation system, also require a high abundance or require the high sensitivity or the sensitivity to be improved. So the goal of my research path over here is to integrate a method that allows us to amplify the signal from mass cytometry analysis. What I did, or what we did as a team, is that instead of labeling the antibody directly with the metal polymer that can chelate the ions or isotope ions, label the antibody first with a small DNA oligo. And then we use the antibody to label or to target the protein or protein phosphorylation site inside the cells or on the cell surface. And this antibody, or a lot of antibodies, are actually fixed in the cells post-staining. And after that, we can play around with the cell with a lot of procedures to do the signal amplification. So what we did is that we flush in extender strands, that is A prime, A prime, and just repeated sequence of the complementary strand of the initiator that you labeled and the antibody. At a low temperature, this extender will fall onto the to hybridize to the initiator, and then with the help of the polymerase BST, it's gonna extend the initiator into a double A structure, which become fully complementary to the extender. And then you increase the temperature in the system, allow the extender to fall off from the initiator strand. Now you have a double A sequence compared to a single A before the process. And then what you can do is just to repeat the process for multiple cycles, driving up the temperature down and up for many different cycles, and then you can generate a really long concatemer or extended initiator sequence with hundreds of copies of the barcode A. And then we have the detector strand or detection strand that is conjugated with the metal that can be hybridized to this, the detection strand binding site, which is just the A sequence, to generate a really high signal or much more increased stoichiometry between the metal and the antibody to allow you to have more abundance of detection agents per antibody. And what is also really critical is that we use a modification called CMVK to modify the detection strand, which can be activated by a short UV exposure to get cross-linked into the concatemer or DNA amplification structure. So after that, everything you see in this structure from the metal to the antibody is connected by the covalent bonding, so that can help the structure to survive a harsh introduction environment in the site of analysis. So yeah, basically using the short oligo, it eliminates non-specific bonding, and using the CMVK-based amplification, the amplification structure can be stabilized and it facilitates the mass spectrometry analysis. So we did many of different type of validations, and this is one of these I'd like to talk about. Is we just developed a transient transfection model where the GFP protein is transfected into a group of millions of cells to create an expression gradient. You can have the cell expressing nothing, so expressing low level, high level. Then we use the anti-GFP antibody to target the GFP, and this antibody is conjugated with A's that can be amplified. And the secondary antibody targeting the primary antibody is conjugated with conventional method that we can use that two signal to compare whether this amplified signal still have the good correlation with the conventional signal. So this is what we're supposed to see. Everything should be, the new approach should be nicely correlated with the conventional approach. But if we see something different than that, that means something wrong, that we can keep pushing the experiment or analysis to a better quality. So this is how what we achieved after the final optimization. We found that the secondary antibody by conventional labeling is really correlated with the primary antibody with A's amplification. And depending on how many cycles you put into the system, you can have a like increased signal, and the correlation is always really good. So overall, we have 13-fold of signal amplification, and in a lot of cases, this is still not enough to read out some really low abundance proteins. Then we can further apply the branching system to allow us exponentially increased signal by like creating this branching tree structure. With this platform, we can achieve over 5-time increase for every branches over here. that allow us to read out the the proteins in single bacteria cells, such as E. coli. And yeah, and another really critical thing for our analysis, because we're doing multiplex analysis, so our method also need to be multiplex. So in the world of DNA technology, the way to do that is by designing orthogonal DNA probe. So we did that, created 50 different probes that can, in theory, to be applied together in the same experiment, and we did it on 33 different probes for experimental validation, and found out actually most of them have really good readout, and only 2 of the 33 strands have more than 10% of spillover or crosstalk, and those ones need to be used with caution. All the others are really good to be used in our system. So yeah, right now is the application. We try to solve this problem one by one. For low abundance proteins, we apply it into EMT system. So I'm not going to the detail of the system, but basically this is the cell model that can go in through EMT and be reversed by going by an MET process to epithelial cells, and we harvest the cell at different time course, at different time points over nearly a month of treatment, and trying to analyze the key phosphorylation proteins or surface markers, phenotypic markers, and low abundance transcription factors such as ZEP1, which shows a really nice trend in this then build up pseudo-time trajectory that can nicely reflect all the other markers that are indicating the phenotypical or phenotype of the cells. So yeah, for low abundance transcription factor, this is a really nice approach to use to allow the amplification. And for signaling network analysis in smaller cells, like primary human T cells, we can also use our methods to amplify the signal, increase the phosphorylation from the basal unamplified level by 17-fold, more or less 17-fold, to really allow the tracking of the TCR phosphorylation network in the time course spanning through 1 hour of after TCR stimulation. And you can see the cells are like the phosphorylation target, like CD3 zeta and ZAP70, that are next to the receptor. These are the proximal signaling response at a really sharp peak, and at the 0.5 or 2 minutes, and they start to decrease. And there's distal signaling network like metric ERG, and will gradually growing up, and they reach their peak at 30 minutes, or sometimes between 15 to 30 minutes before they start to go down. And internal control, phospho-SMAD2, which is not relevant to this the network, is still stay the same, like indicating our method to be really specific. And we can use that to build up the network, and we found the distal signal is more sustained compared to proximal signal. And lastly, we can also use that for imaging mass cytometry, which is an alternative technology that's replaced the sample introduction system with a laser ablation, so you can directly read out from the tissue slice, but it has 1 micrometer square resolution. That means it contains a much less sample material compared to a single cell. That's you need higher degree of signal amplification on this method for sure. So we modify our approach to make it the cyclic amplification, or compatible with tissue slice. And we do that on 20 tissue-specific markers on human kidney tissues, and found out like every single one of the markers just been light up with great signal that allow us to subclassify those cells and put them into different clusters that are blood vessel, glomeruli, and different tubular structures. And within each cluster, there's also a high degree of heterogeneity, which can be analyzed and put back into the original plot to analyze where the heterogeneity is coming from. So yeah, I would like to end. give a summary, which is that we apply DNA technology with the mass cytometry to enable the analysis of low abundance marker at the single cell resolution. And yeah, so this is where the lab, Dr. Peng Yin's lab, where I did my postdoc, where this method is developed. And this is my new lab at the University of Minnesota. We're still small, but we're trying to use this method and also develop many varieties of different approaches to help with the single cell proteomics analysis. Thank you.
I think Jonathan, great talk, by the way. Jonathan Smealer made a point during the panel discussion about needing lots of cells, and certainly this is a method for that. So what types of applications do you think having this like much greater number of cells is kind of best for that we can try to leverage and combine with data that other types of methods produce?
Yeah, I think there's a like different, it's really kind of complementary to have different approaches for single cell mass cytometry. We don't have the coverage as big as the proteomics, but we have a good throughput, and the quantitativeness is also like good. And also, yeah, there's a I think our goal, or our also part of the future research line from our lab, is trying to use that as first of all as a screening method, so we can analyze that in like as a screening approach for drugs for different combinations of perturbations. We can use that to profile the human sample, patient sample at a really good speed. We can analyze like millions of cells from one patient times like tens or hundreds of patients in a single days of experiment to get a lot of information, especially when there's like high degree of heterogeneity. And yeah, and I think also although we don't have a really good multiplexity as what you guys have, but there's different ways that we can incorporate DNA markers, RNA, protein, PTM in a single run to generate this kind of multi-omics or multi-parameter analysis. So this is what we trying to do.
Maybe just another question here. So I think the temporal component, being able to analyze these signaling networks, is really exciting. Are there other types of signaling networks beyond kinases and phosphatases that you're particularly excited to apply this technology to?
I think the main problem is that for any signaling analysis, we need to have good antibodies that can target specifically that particular phosphorylation site on the particular protein, and this is really achievable with phosphorylation. But for a lot of other PTMs, you just don't have the possibility to read out that with this high degree of specificity. But also we are developing other approach, or using DNA probes to try to achieve that by using for example, two antibodies, one bind to the phosphorylation or a pan-phosphorylation or pan-PTM target, and another antibody bind to the total protein. And then these two proteins can bind to the same target and create a reaction, generate a record when that particular PTM happened on that protein. It starts to record and generate signal, and this is how we try to achieve that and apply that on different kind of PTM.
Great. Well, if there are no further questions, let's give a round of applause to Xiaokang.